Hi, good evening, all of you. So, I know that you are all on the last and final lap of your preparations. And in March 19th, Tuesday, we are waiting for our day. And the last 1 year, we were all, working very hard, right? Okay, so there are 13 chapters. 13 chapters are there in our textbook in NCRT, and this is the final day or this is the final lap of preparation, and we are going to execute whatever the things you studied in the last year. Everything you are going to perform well in March 19th. Okay, so it is the chance to show your brilliance. Okay, so that's why I'm without wasting the time, I'm directly moving into the topic. And today, and as you all know that, my dear students, you know that the total 5 units are there in biology. And among the 5 units, the most important unit is nothing but genetics. You know that. So genetics and evolution, one of the important unit by which you can see there are two chapters are purely related with genetics. One is principles of inheritance and variation, and the next one is molecular basis of inheritance. And I think you know the weightage of the chapters. There are two chapters. Each chapter is nearly 8 marks. So 8 marks means 8 plus 8, and that is 16 marks are there from the topic principles and molecular basis. And most of the students are struggling about some of the important areas in this chapter. Especially in principles of inheritance and variation, there are some disorders are there, especially the genetic disorders. And there are two different types of genetic disorders, as you all know that. What are they? One is called what? The Mendelian disorder, and another one is called the chromosomal disorders. Okay, and students are having some basic confusions related with these two topics. Anyway, so we are doing the Excel Educational Center, and we are doing the live related with and not some tricky questions. Okay, so if you are discussing some one or two tricky questions, and that will not be helpful for you, and the reason is, and you need a complete revision of the whole chapter. That is the thing that you need now, my dear students, because almost you covered the whole chapters, and there are 4 more days waiting. So that's why I'm not concentrating on the important as well as the difficult topics, my dear student, instead. And my point is nothing but we are covering almost the whole topics from the textbook. Okay, so that is why the reason. Please be ready for the class, my dear students. Okay, so we can start. You know that the first chapter, principles of inheritance and variation, you can see the first chapter. Listen, you can see the first question. And what is the first question? And here you can see it is a question related with the introduction of the topic itself. These two chapters, I already told it is something related with the genetics. Genetics is the study of genes, that. And as you all know that when we are discussing the genetics, you can see where is the gene located. Okay, so where is the gene located, where is the DNA, where is the chromosome, and the nuclear content, chromatin, nucleosome, heterochromatin, euchromatin. So altogether, almost all the things what I told, everything is there inside the nucleus. So the discovery of electron microscopes revealed the secret behind biology. Okay, and before the discovery of electron microscopes, you can see the biology, there are so many unanswered questions. Biology is actually full of unanswered questions. But the discovery of electron microscope, and you can see, and that made a very, very important milestone in the field of biology. So electron microscopes means what? We can see what are the things located inside the nucleus and what is happening inside the nucleus, inside the DNA, inside the gene, and including inside the chromosome. So that's why the reason recently, the last, I think the 4 years back, we were completely locked down due to a particular virus called COVID-19. And after COVID-19, whatever the, what we can say, the researchers as well as you can see the scientists fully involved to find out a vaccine, and that is the RNA vaccine, because as you all know that COVID-19 is a retrovirus. COVID-19 is a virus with RNA as a genetic material. So that is why reason you can see the biotechnology, it's a branch of genetics, you can see, and that is highly, highly advanced, and finally we made some solutions. And only due to the solutions, my dear student, we people are alive today. So that is the reason, that is the advancement in the field of genetics. So here we are going to discuss some important areas from the first chapter, principles of inheritance variation. Okay, so the first question, my dear student, and what is that? The gene responsible for. So you know that whenever we start a topic with genetics, and we should remember one of the eminent personality is nothing but Gregor John Mendel. Gregor John Mendel, considered as the father of genetics, my dear student. He was the person who laid a foundation stone in genetics. So he conducted so many experiments, and as you all know that what is the experimental plant, that, and it is the garden pea plant. And what is the scientific name of garden pea plant? And it is Pisum sativum, my dear students. The garden pea plant, the scientific name is Pisum sativum. And the word is not there in the textbook, but in your textbook that word is there. Which one? Garden pea plant, the word is there, but the Pisum sativum, that word is not there. But whenever there is a question related with the experimental plant of Gregor John Mendel, you must write the word Pisum sativum. It's a scientific name. When you're writing the scientific name, there should be some rules, because that is our universal rules. That when you're writing the first one, this is the genus name, this is the species name, and the genus name should start with a capital letter, and the species name should start with a small letter. When it is handwritten, that should be separately underlined. And here I look at, and here I underlined it separately, and the first letter is capital letter, all other letters are a small letter. Pisum sativum, the experimental plant. It is having a very short plan. It's having very short life cycle. Within 3 months or 4 months, you can see they are developing the new generation. And at the same time, why Mendel selected the garden pea plant for his experiments? There are so many criteria. The first one, the nothing but the most important one, that is nothing but you can see it is having a short lifespan. Okay, and it is very easy to handle, and it is a bisexual flower. And you can see what is easy to handle. That artificial hybridization you studied in another chapter. What is artificial hybridization? And taking the plant and removing the male reproductive part. And all of you can remember what is that process of removal of that male reproductive part of a flower is called what? Emasculation. Emasculation. And after emasculation, we are bagging it, and after that we are dusting it, and again we are doing the rebagging, and that is what we call artificial hybridization. And artificial hybridization is also one of the important steps conducted by Gregor John Mendel. And the most important thing here, you listen, my dear students, here you can see the question is related with Gregor John Mendel. The question is related with the garden pea plant, and the garden pea plant. And what about the total? Sometimes when you read the question, you can see the question is not familiar to you, I think. Because you can see the garden, when we are taking that plant into account, and how many chromosomes are there, my dear student? How many chromosomes are there? The pea plant, the garden pea plant is having how many chromosomes? That is very, very important. How many chromosomes we have? We have 23 pairs of chromosome. Why we are not using the term 46? Because you can see, and each one is homologous pair, and that is one of the most common word in genetics. That. So the first chromosome, as you all know that if this is the first chromosome, this is the second chromosome. The number of genes in the first chromosome is equal to the number of genes in the second chromosome, and that's why the reason both are having the same shape, same size, the same number of genes in same location. All together, we can use a term homologous chromosome. If this is the first chromosome, this is the second chromosome, both chromosomes are pairing. That is what we call homologous chromosomes. So homologous chromosomes are paired. So that's why the first chromosome and second chromosome is paired as first homologous pair, third and fourth paired as the second homologous pair, and the fifth and sixth to pair to form the third homologous chromosome. So all together you can see 23 homologous pairs you can see in humans. And and the question is not related with the humans, that the question is related with the pea plant. How many chromosomes, my dear students? And please, if possible, please give me the response through the chat box, my dear students. Okay, so then only the classroom will be an interactive one. Okay, and here you can see the pea plant, you can see the total number of chromosomes. There are 14 chromosomes are there, and that a term is also not given in the textbook. 14 chromosomes are there, and out of the 14 chromosomes, there are 7 pairs of chromosomes. 7 pairs. So 7 pairs means what? 14 chromosomes, right? And 7 homologous pairs. And here, the Mendel conducted so many experiments in garden pea plant. How many characters, how many traits selected by Mendel, my dear students? How many characters, how many traits selected by Mendel? That also you are very familiar with. That is 7. As you all know that here you can see, and this is the picture you can see, my dear student. And there are 7 characters selected. And Mendel conducted experiments in 7 years. And after that, in 1865, he published all his findings in his book. And unfortunately, all his findings get totally rejected, that. And you can see what the main issue is, nothing but the communication issue, my dear students. Nowadays, you can see the communication is highly, highly advanced, right? That's why the reason, I am taking class over here and you are getting everything in your fingertip. And in 1865, you can see the communication system was not well developed. So that's why you can see most of his findings were totally get rejected. And especially you can see the term given by Mendel is nothing but factors. That he introduced a term named factors. The parents are having factors, and the factors are transmitted to the next generation. And now in the modern world, we have no idea about factors. And instead of factors, what we are using the term? We are using the term DNA. We are using the term chromosomes. We are using the term genes, genetics, the modern genetics. So here you can see, when we are looking that serial number, there are 7 traits are there, and the 7 traits are also given here. Stem height, you can see there are 2, tall and short, and here tall and dwarf, you can see flower color, flower position, and flower color violet and white, flower position axial and terminal, pod shape, pod color, seed shape, seed color. These are all very, very simple to you, I think. But the thing is, what is the last column? That last column is new to you, my dear students, because we are not familiar with that. I already told how many chromosomes? 7. 7 pairs of chromosome, and 7 pairs of chromosome means what? What is the distribution for stem height? You can see a gene is there. No, a gene is there for stem height, and it is not one gene. One pair of gene is responsible for expressing a character that you already studied. No, what is that? And a gene, a single gene cannot express. And when a pair of chromosome, that also you studied a theory called what? Chromosomal theory of inheritance. The concept chromosomal theory of inheritance was given by Sutton and Boveri in 1902. And the chromosomal theory of inheritance, as you all know, that the chromosomal theory of inheritance is always giving an idea about the parallel behavior between the gene and chromosome. The chromosome and genes are showing a parallel behavior. That parallelism is given by the Sutton and Boveri in their theory, chromosomal theory of inheritance. So here you can see 1 pair of gene is there, right? And for stem height, 1 pair of gene, flower color, 1 pair of gene, flower position, pod shape, and where is the gene? That is very important. Here you can see, we can take one by one. The first one, you can see the first one, the first chromosome. There are, look at the board, my dear, the first chromosome, you can see the flower color and seed color. The flower color and seed color, both are there in the first chromosome. How many chromosome? 7 chromosome, 7 homologous chromosome. And the flower color and seed color, both are there in the first chromosome. And what is the second one? And nothing is there in the second one. There are so many characters are there in the second chromosome, but Mendel studied only 7, so that's why no such characters he studied from the second chromosome. And the third one, there is no third one, but the fourth one, you can see 1, 2, 3. The fourth one, the height is there in the fourth one, and you can see the flower position as well as pod shape. These are the 3 characters, my dear student, in chromosome number 4. And in chromosome number 5, there is only 1 character. Chromosome number 5, there is only 1 character, the pod color, my dear student. And chromosome number 7, the last one also, there is only 1 character, seed shape, round and wrinkled. So all together you can see all the 7 characters are distributed. All the 7 characters are distributed among 7 chromosomes. And the 7 homologous chromosomes. And and what is the question here? And here you can see the gene responsible for flower color. And the first one, look at the board, the flower color, my dear student. Flower color is there in which chromosome? The first chromosome. The flower color is there in the first chromosome. Here you can see what is the flower color. This is the flower color. Flower color is there in the first chromosome. And here you can see this is the flower color. And where is the height? You can see height is there in the 4th chromosome. And here you can see the pod color. Pod color is there in the 5th one. And you can see the seed shape in the last chromosome. We discussed no seed shape in the last chromosome. So this is the right answer. At least you have to study this point. What is that? And the Gregor John Mendel conducted experiments in garden pea plant named Pisum sativum. And that Pisum sativa in the garden pea plant consists of 7 pairs of homologous chromosomes. And out of the 7 pairs of homologous chromosomes, chromosome number 1, chromosome number 4, chromosome number 5, and chromosome number 7 are the 4 chromosomes by which the 7 characters are involved. At least you should be familiarized with that these 4 numbers: chromosome number 1, chromosome number 4, chromosome number 5, chromosome number 7. Okay, that's very important. And whenever you are getting a chance to write this point in the board examination, you can add this point as an additional point, my dear students. Okay, and that's why the reason you must have an idea about how many chromosomes in total and what are the chromosomes by which Mendel selected. I hope the first question is clear. Moving into the next question, my dear students. So, when we are discussing the chapter itself, you can see topic-wise we are discussing. So, the first one, the Grigor, John Mendel, the introduction, but we can say the characters, traits, everything. Okay. The second question, in a typical Mendelian dihybrid cross, you know that the Mendel cross, mainly there are 2 different types of crosses he conducted. One is called the monohybrid cross, another one is called a dihybrid cross. And here in this question, and what is the secret behind that question? In a typical Mendelian dihybrid cross, they asked a total 560 seeds are produced in F2 generation. Oh, it's an important question, my dear student. And in a textbook, you can see the number of seeds are not given. Hey, whatever the number of seeds produced, what is the ratio you studied? What is the ratio? What is the ratio? If the ratio is 9:3:3:1 is the ratio. So you can see the Mendel conducted monohybrid cross and dihybrid cross. In monohybrid cross, what is the ratio? And what is the ratio in monohybrid cross? And it is 3:1 as well as 1:2:1. Why 2 ratio? 3:1 is the monohybrid phenotypic ratio, and 1:2:1 is the monohybrid genotypic ratio. And here you can see what is 9:3:3:1. 9:3:3:1 is the dihybrid phenotypic ratio. So 9:3:3:1 means what? How many total boxes are there? When you are doing a dihybrid cross, you have to draw a Reginald C. Punnett developed a Punnett square. You can see in that Punnett square, you studied nearly 16 squares. You can see in the 16 squares, you can see that is distributed in the form of the phenotypic distribution is nothing but 9:3:3:1. And here the total 16 boxes you can see. And what is the genotypic ratio? Genotypic ratio is not that much important, but you have to study that. It is easy, my dear student. 1:2:1 is the ratio of monohybrid, right? So you have to write first monohybrid ratio 1:2:1, and again you have to double it because dihybrid, right? Double it. So 1:2:1, 1 double 2 and 2 double 4 and 1 double 2 you can see. So 1:2:1 and just double 2:4:2. Then again you have to write 1:2:1. And this is the ratio, my dear students. And this is the ratio, the genotypic ratio, the genotypic Mendelian ratio. Which one? Dihybrid ratio. So 1:2:1, 1:2:4:2, 1:2:1. I will repeat one more time. 1:2:1, 2:4:2, 1:2:1 is the Mendelian dihybrid ratio. And here the question is related with the Mendelian phenotypic ratio, my dear student, because the question is, in a typical Mendelian dihybrid cross, 560 offsprings are produced. So the question, 560 offsprings are produced in the F2 generation. And in F1 generation, there is only one type of young one is there in F1 generation in all typical monohybrid and dihybrid cross. But you can see in F2 generation, in monohybrid, there are 2 different types of phenotypes. And in F2, a dihybrid cross, you can see there are 4 different types. And here, 4 different types means what? The 9 is parental, my dear students, as well as the last one is also parental. And you already studied one law related with the dihybrid cross you studied. What is that? And along with the parental, parental, you can see the non-parental. That term is very important, my dear student. Along with the parental, not in monohybrid. Monohybrid only parental. Monohybrid only parental characters. And what about dihybrid, my dear student? Dihybrid, and you can see the dihybrid characters. You can see here. You can see along with the parental, there are some non-parental characters are also produced. So non-parental characters are produced only in dihybrid cross, my dear student. So here you can see the 9 is parental, the most one is parental, least one is parental, but the intermediate one that is non-parental. How the non-parental young ones are produced? That is due to the law of. How many of you can write that one? It is the law of independent assortment. Important question for the exam. Independent assortment. Law of independent assortment. What is law of independent assortment? When two pairs of characters, law of independent assortment is starting with when two pairs of character. Two pairs of character means what? It is not applicable in monohybrid. Monohybrid is always dealing with what? Mono means one, only one pair of characters. But here you can see dihybrid means what? When two pairs of characters. This is one pair of character, this is another pair of characters. When two pairs of characters are, and when two pairs of characters are segregated. When two pairs of characters are segregated, you keep in your mind one more thing. What is this one? This is first pair of character, this is the second pair of character. When these two pairs of characters are undergoing segregation. What do you mean by segregation? What do you mean by segregation? Aggregation means joining. Segregation means separation. Opposite word of aggregation, that is segregation. And during the segregation, look at my hand, my dear student. This is one pair of character, this is the second pair of character, and the segregation of the first pair is independent of the segregation of the second one. Hey, that is a independent. I used a word independent. What is that? Segregation of the first pair is independent of the segregation of second pair. That is the key behind that particular reason, my dear student. That is the law of independent assortment. And here you can see the law of independent assortment. So that's why due to that independent assortment, along with the parental, non-parental characters are also produced. But how can we find out the answer? How can we find out the answer, my dear student? And that is also very important. What is the ratio? I told 9:3:3:1. When there is a question, how can you solve this question? All of you look at the board, then all you get the idea. Please listen. In a typical Mendelian diabetic close, 560 offsprings are produced in the F2 generation. How many offsprings will approximately be phenotypically non-parental? The question is phenotypically non-parental and phenotypically parental. Okay, so first of all, we should get the idea about how can we solve the problem. Listen, if the question is 560, and first of all, you have to do that 560, how many phenotypes are there, my dear student? There are 4 different phenotypes, 9:3:3:1. And at the same time, how many boxes are there in F2? 16 boxes, so that's why 560 divided by 16. Because if it is a monohybrid, you have to divide it with a 4. Because that why divide it with 4? Because there are 4 boxes are there. And here 560 divided by 16, and 560 divided by 16, that is 35. And 35 is the answer of 560 divided by 30. And that 35 multiplied by 9. That 35 multiplied by 3, that 35 multiplied by this 3, and that 35 multiplied by this 1. That is the secret behind that value, my dear students. How can you do the problem? If it is a dihybrid, in F2 generation, how many seeds you got? Here I told why 560, and in exam there may be chance for asking different numbers, more than 1000 or 2000 or 3000, whatever the number, you have to divide it by 16. So you'll be getting an approximate value, and it is not an exact value just like mathematics, but here you can see an approximate value is there, because after conducting an artificial hybridization, we are getting the number of seed means what? Number of seeds are not a mathematical number, my dear student. So that's why instead of 560, even if it is 561 itself, it is considered as what? 560 itself. Okay, and here you can see the 35, and when you are multiplying with the 35 into 9, 35 into 9, you are getting a value, right? 35 into 9, that is 315. Okay, so 35 into 9, 315. That means what? 315 offsprings are parental. And what is this one? And that is what we can say, that is 35, my dear students, 35. Hey, why? 35 into 1, 35 into 9, 315. So altogether you will be getting 350, my dear students. And 350 young ones are parental. Because one is 9 parental, right? And one is, I think it is clear, or otherwise we can use an example, then you will get the idea. Please, okay, look at the board. Here you can see, for example, when you are crossing a tall into what we can say the seed shape, height into seed shape, and here you can see what is that? 9, and 9 will be tall comma round. Both are dominated, tall comma round. What will be the least one? And tall means what? And here dwarf you can see round, instead of round you can see wrinkled. So how many tall and round? 315. How many dwarf and wrinkled? 35. So 315 plus 35 together, 350 parental characters. And what is remaining? And 35 into 3, that is 105. And 35 into 3, 105, my dear student. So that's why 210. So you know the 105, 105, 305, 305, and you will be getting 210. And when you are adding these two, and that will be the 560. That is our value, my dear students. This is the value. 560 is the value. So out of the total number of 560 seats, out of the total 560 seats, you know that 350 seats are 350 seats are parental, and the remaining 210 seats are non-parental. These parental and non-parental are applicable only in dihybrid. And what about if the question is a monohybrid clause? You can say that all are parental. Why? If 560 seats, all are parental. Why all are parental? Because in monohybrid clause there is no non-parental character. So that is the difference. So this is the method to solve the question. So how can you solve the question? I will repeat one more time. What is that? How can you solve the question? If it is a monohybrid clause, divided by 4 into ratio. And if it is a dihybrid clause, divide by 16 into ratio. So when you are in a monohybrid clause, the number divided by 4 into 3:1 ratio. If it is, if they ask about the tall, it is 3. If they ask about dwarf, 1. Okay, and if it is a dihybrid clause, you have to divide the number with the 16 and multiply with the ratio. What is the ratio? 9:3:3:1. The 9 you are getting the parent, the dominant parent, and 1 the recessive parent. In between these two, the mixture of dominant and receive and dominant. Here dominant. You know that you studied. This is dominant. This is recessive. Dominant is the parent. Recessive is the parent. What is the non-parental one? Dominant, recessive. What is the next one? Recessive, dominant. What is the parent actually? Dominant is the parent. Recessive is the parent. Then what will be dominant recessive? Non-parental. What will be recessive dominant? That also non-parental, right? And the question is, how many offsprings will approximately be phenotypically non-parental? They use a word approximately. I already told you it is not a mathematics number, so that's why approximate number. It is 350, my dear student. And there is one more question is there. How many of them are phenotypically parental? And the phenotypically parental one is 350, and the phenotypically non-parental one is 210. And when you add both, you will be getting 560. It's an important question, my dear students. Okay, so I already told you, out of the total percentage of marks in biology, and 20% of the questions are creative. 20% of the questions are higher order thinking question. Okay, analyzing question, and the remaining 80% of the question either average or below average. But you can see the 20% of the questions are always tricky, my dear student, and the 20% of the questions and will determine your position in the first rank, my dear students. So that is why the reason that 20% of the questions are also very, very important. Without wasting the time, moving into the next question. There is a question, what is the genotype? So the topic wise we are discussing, what is the genotype and percentage of pink colored flower in F2 generation of Antirrhinum majus? I think you are very familiar with that plant, Antirrhinum majus. This is a scientific name. But what I told in the beginning, when you write a scientific name, that should be separately underlined. It is not separately underlined. Why? Why it is not separately underlined? There is a rule. What is that? When it is handwritten, only separately underlined. Is it handwritten? No. It is printed, right? When it is printed, it should be in italics. And you can see it is in italics, my dear student. You can see it is in italics, Antirrhinum majus. Okay, when it is handwritten and in examination, you can do write it the word right. So that's why you have to separately underline. But here in the printed form, it is italics. Okay, so Antirrhinum majus, what is the plant? Antirrhinum majus, Mendel developed, 3 important rules, that 3 important laws, that one is called the law of dominance, other one is called the law of segregation, and the last one we just mentioned, that is law of independent assortment. What is the first law? Law of dominance. Unfortunately, the first law of Mendel is totally rejected by the world. Why the first law, the law of dominance, is rejected? And there are so many evidences are there against the mentalism, my dear student, against the law of dominance. And what is law of dominance? One dominant allele can completely dominate over the recessive one. That is called what? Law of dominance. But here, the mental postulated the law. One dominant allele can completely dominate over the other one. But what happened actually? And there is a phenomenon came. You are very familiar with that. What is the phenomenon? Please, what is the phenomenon? Here you can see it is called incomplete dominance. It's a very important, my dear students, incomplete dominance. And that concept is very important, my dear students. In your textbook, there are two examples are given. One is starch content in the pea plant. Most of the students are writing first example means what? They're all concentrating on anti-rhinum majors, the snapdragon plant. And you can see the dog flower. In that plant, you can see, what is that? The red colored flower crossing with the white colored flower. Which one is the dominant one? Red colored one. So capital R, and here small r. This is not the cross they asked, and this is the cross by which you can see both phenotypic and genotypic ratios are same. Both are 1:2:1, 1:2:1. What about Mendelian ratio? Mendelian ratio is 3:1 and 1:2:1. And both are 1:2:1 means what? And it is something unique, right? And here it is incomplete dominance. What is incomplete dominance? And here you can see when we combine capital R, small r means what? And if you apply Mendelism, if you apply the law of dominance here, capital R can completely dominate over small r, and that will be producing the red color. But here you can see capital R, my dear students, capital R cannot dominate. Capital R cannot completely dominate. Capital R cannot completely dominate over small r. So that's why which color will be produced, my dear student? Pink color is produced. According to Mendel, either red or white. According to Mendel, either red or white. Only parental in monohybrid. Instead of parental, a non-parental character is expressed in the F1 generation itself. That is the pink color. So that is why the reason that is considered what? Against the law of dominance. That is called incomplete dominance. So whenever there is a question in incomplete dominance, how many examples are there in your NCERT textbook? One is incomplete dominance in anti-rhinum majors, red and white to form pink. And pink, when we are doing the cell pollination, we are getting 1 red is to 2 pink is to 1 white, 1:2:1, or 1 capital R is to 2 capital R, small r is to 1 small r, 1:2:1 ratio. But this is one example. What is the second example? Starch synthesis in pea plant is also an example of incomplete dominance. What is starch synthesis? Some plants can make large quantity of, means what? That is round shaped, and other ones wrinkled shaped. In between an intermediate shaped seed, that is due to incomplete dominance. These are the two examples given in your textbook for explaining the concept incomplete dominance. Here you can see what is the question. What is the genotype? What is the genotype of the pink? Hey, I think you can easily give me the answer, my dear student. What is the genotype of the pink? That is capital R, small r, my dear student. What is the genotype of the pink? That is called capital R, small r. And here one more question. What is the percentage, my dear student? What is the percentage of pink flower in F2 generation? And what is the ratio? 1:2:1. What is the percentage actually? And the 1 means what? This is 25%. What is 2 means what? 50%. And what is this 1? That is 25%. So 25% red, 50% pink, 25% white. And the question is, what is the percentage of pink? The answer is 50%. Chance for asking an MCQ type of question, 50%. Okay, so there are two questions are there in that. One is the genotype of the pink, and other one percentage of pink. The genotype capital R, small r, and the percentage, it is 50%. If the question is red, 25%. If the question is white, 25%. 25% plus 50% plus 25%, that is 100%. And that is 1:2:1, both phenotypic and genotypic. Next question. So there are 5 questions are there, my dear student, coming under name the following. That is an easy topic, my dear students, because you can see when we are doing this question, I can cover most of the portions from the textbook, my dear student. Name the following. Okay, so I think you are all giving proper response, my dear students, please. Okay, and here the name the following, the first one. What is that? The codominant human blood group. And codominance, you studied, no? Codominance, what is codominant? You can see what is codominance actually. Means you can see when two alleles, they are heterozygous, come together, neither the dominant allele or neither, what we can say, the first allele nor the second allele can dominate over each other. Neither nor. You must use the word neither nor. Neither the first allele nor the second allele can dominate over each other. And you studied, human blood group is the best example for multiple allelism. Human blood group is one of the best example for multiple allelism. What do you mean by multiple allelism? Multiple allelism means what? Here you can see a character is determined by more than two alleles. That is what we call what multiple allelism. And multiple allelism, the blood group, you can see what is the allele. You can see what is the allele. Come on, what is the allele? Here the allele is, listen my dear student, it is what we call what? The I gene. The human blood group is determined by a gene called I gene, and I gene there are three different types of allele. One is called IA, other one is called IB, and the last one is called what? Small I. And you know in some textbook it is given IO, no problem, both are correct. You can see IA, IB and small I. IA and IB are dominant alleles, while small I or IO is, you can see, the recessive allele. And here using this one, you know there are different combinations are there. And here the question is, which one is the codominant blood group? What do you mean by codominance? I already told. No, what is that? Neither the first allele nor the second allele can dominate over each other. So that's why when IA and IB, you know, our blood group is determined by the presence of an antigen in the surface of RBC, and that antigen is produced from the genes from our parents, and this is the paternal gene. Paternal gene means what? The gene which is given by your father. And what is the second one? The maternal gene, the gene which is given by your mother, and this is the paternal gene. This is the maternal gene. Father is giving IA, mother is giving IB. When both comes together, it is heterozygous condition. In heterozygous condition, what is the rule of Mendel? What is the law of Mendel? What is that? And one allele can dominate over, one allele can completely dominate over the other one. Unfortunately, that law of dominance is not applicable here. Here, IA, neither IA, instead of first gene you can write IA, neither IA nor IB can dominate over each other. So that's what will happen. IA or IB are not dominating over each other means what? Both are expressing together. Both are expressing together means what? And that is why, and we are having a blood group called what? AB blood group. Otherwise, either we are having A group or B group or O group. And here A group, the normal law of Mendel. B group, the normal law of Mendel. And you can see AB group is a theory against Mendelism. Against Mendelism. That is what we call what? Codominance. What is codominance? You know, cooperation. And you know, neither IA nor IB can dominate over each other. That's why you can see both are expressing together. IA is expressing to make A group. IB is expressing to make B group. We are having a blood group called AB group, the universal recipient. AB is a universal recipient, right? And O is the universal donor, right? The universal recipient blood group is why they're called universal recipient. Grade 11 topic, right? And it is not having any antibodies in plasma. Okay, so you know I'm not moving into that. So what is the question? And the codominant human blood group, it is AB group. So AB group is called the codominant blood group. There is only one group, AB group. Okay, what is the second one? The scientist who gave experimental verification. Oh, that is very important, because whenever you read the word chromosomal theory of inheritance, whenever in the question paper, when you read the word chromosomal theory of inheritance, all are having a particular scientist name is coming into your mind. That is nothing but what? Sutton and Boveri. You studied Sutton and Boveri introduced chromosomal theory of inheritance. But the question is, who proposed chromosomal theory inheritance? Okay, your answer is correct. But here the question is, who gave the experimental verification? And you know, the theory and experimental verification both are very, very important. In the chapter molecular basis, you studied, DNA replication is semi-conservative. What do you mean semi-conservative? What is semi-conservative? A parental DNA divided to two daughter DNA. Each daughter DNA, one is parental strand, other one is newly synthesized strand. And you can see half of the parental strand is conserved, half semi-conservative, conserved. Who proposed that one? Who proposed that concept? DNA replication is semi-conservative. It is nothing but James Watson in 1953. Along with, they proposed a concept, right? What is the concept? Watson and Crick, you can see James Watson and Francis Crick together proposed double helical model of DNA in 1953, my dear student. The same year, 1953, James Watson and Francis Crick together proposed the DNA replication is semi-conservative. But the question is, who gave the experimental verification of semi-conservative? It is not Watson and Crick. It is Michelson and Stahl. You study that experiment, Michelson and Stahl experiment. Name of the scientist, very, very important. And here the question, the scientist who gave experimental verification of chromosomal theory. Chromosomal theory of inheritance was given by Sutton and Boveri, but you know the concept was experimentally proved by Thomas Hunt Morgan. No need to write the full name, at least you have to write T.H. Morgan. T.H. Morgan, Thomas Hunt Morgan. And you know, T.H. Morgan conducted a lot of experiments in one of the very simple insect you study. No, what is that? The fruit fly. Do you have any idea about the scientific name of fruit fly? Because you must write, your biology students, you must write the scientific name of fruit fly along with the word fruit fly. That is Drosophila melanogaster. So Drosophila melanogaster, you must write Drosophila melanogaster or fruit fly. And you know the person, the eminent personality is T.H. Morgan, the Thomas Hunt Morgan. Okay, moving into the next one. Name the following means what? You have to write the answer only. No need to give any other extra point. For 3 marks question and 5 marks question, you have to, that is explanation, you know, it is what we can say explanatory or what we can say descriptive type questions. Even you can see the MCQ, you can write the answer only, and you can see the 2 marks question also, you have to point out the important points only. But 3 marks and 5 marks, you know, if you write only points, you will not get the mark because they are descriptive type of questions. And you know, in the question paper, in the first, before starting the first question, there are some general instructions. Most of the students are not reading that general instruction. And what is the last instruction in the first page of the biology question paper in CBSE board examination? It is nothing but wherever necessary, you have to draw the proper diagrams. And generally, we are all thinking that word. And if there is a question, what is that? Draw the diagram. No, my dear student. Even if it is not asked itself, it is your responsibility. It is your responsibility to draw the diagram in 3 marks and 5 marks, 3 marks and 5 marks. Okay. And here, because that, you know, already they are given the direction in the first page itself. In the first page itself, it is given. That's why the reason, please. Okay. And here you can see the third question. The relation between linkage and recombination, an important question, my dear student. Okay, and you know, what is the relation? What is linkage, my dear student? What is linkage? In a chromosome, look at the board, my dear student. In a chromosome, you can see if this is one gene and if this is another gene. And here, look at the board. Here you can see this is another chromosome, and in another chromosome you can see, and this is the, look at the board, please. This is one gene and this is one gene. What is the difference between these two diagrams? What is the difference between these two diagrams? Here you can see these first and second genes are located. Linkage. Oh, very high linkage, right? And here you can see the physical association between 2 genes in a chromosome is called what? Linkage. So here you can see these 2 chromosomes, these 2 genes are closely linked. So here the linkage is maximum. What about here? Here you can see these 2 genes are located close, right? And but these 2 genes are located away from each other, right? So that's why the distance between 2 genes, repeat one more time, the distance between these 2 genes are comparatively more than the first case. And here you can see the distance is less, means what? They are closely linked. And here you can see they are located away from each other. So that's why here you can see that this is the first chromosome, this is the second chromosome. Here the linkage is less. And linkage is more. So more the linkage, what will be the recombination? And if the genes are closely linked, the chance for producing new combination or recombination is less. So we can say when linkage increases, recombination decreases. So that's why here, what is the question? The relation between linkage and recombination. Is it directly proportional? No, it is not directly proportional. It is inversely proportional. How can you represent like this? L is inversely proportional to R, you can see. So linkage is inversely proportional to recombination. More the linkage, less the recombination. In the first case, you can see more the linkage, so that's why less recombination. And my dear students, you know the second one, less linkage, so that's why more recombination. Who developed these concepts? You can see, you know, one of the eminent scientists, you can see Alfred Stewart Wand. Alfred Stewart Wand, you can see, conducted so many experiments, and finally, actually, you can see who is the teacher of Alfred Stewart Wand. The teacher is nothing but Thomas Hunt Morgan. Thomas Hunt Morgan introduced the concept linkage and recombination, but unfortunately, the teacher cannot give the proper explanation about the distance between 2 genes. Hey, a teacher is explaining this is the method, but the teacher cannot give what is the distance between the two genes. And one of the student inspired by the teacher, one of the student named Alfred Stewart one, inspired by the teacher, motivated by the teacher, and he was the person developed the concept, the distance between two genes. What is that? One of the important branch of genetics called gene mapping, my dear student. The gene mapping concept was the gene mapping, the distance between two genes. That concept was developed by Alfred Stewart one, topic in NCERT textbook. And at the same time, he in memory of his teacher, he developed the unit, right? What is the unit, my dear students? CM. Whenever we are using the cm for distance, we are commonly using a term centimeter. Is it centimeter? No, it is not possible to use centimeter to describe the distance between two genes. We are using, what is the word? It is called what? Centi. What is that? Cm. If it is both are small letter, that is centimeter, right? If C is small letter and M is capital letter, that's why it is called what? Centimorgan. Morgan is his teacher. Okay, so that is why the reason, what is the unit between, or what is the distance between two genes, that is centimorgan. And that's about that a topic. You can see the relation between linkage and recombination. What is the next one? Question number D. Okay, so D, what is the next one? A gene that can exhibit multiple phenotypes. A gene that can exhibit multiple phenotypes, my dear student. And generally you can see, listen, one gene can exhibit only one phenotype. That is a common thing. For example, melanin gene can exhibit as a melanin protein. So each gene can express one protein, but what is the question here? A gene that can exhibit multiple phenotype. For example, if this is a gene, look at the board, my dear student. This is a gene. This gene is having a phenotype 1, a phenotype 2, a phenotype 3. Is it one phenotype? No, more than one phenotype. If a gene can make more than one phenotype, if a gene can exhibit multiple phenotypes, do you have any idea what is the name of that particular gene? You can see that gene is important, my dear student. That gene is called a pleiotropic gene. Pleiotropic gene. What is that phenomenon called? What? The phenomenon is called pleiotropy, and the gene is called pleiotropic gene. You already studied one example, PKU, phenylketonuria. The phenylketonuria is one of the autosomal recessive disorder, my dear students. Here you can see the phenylketonuria, one of the Mendelian disorder, one of the autosomal recessive disorder. That is an example for pleiotropic gene. Okay, and here there is the answer, pleiotropy. What is the last question under that particular category? Name the following. The phenotypic ratio obtained in the F2 generation in human skin color. Okay, so that is also an important question, my dear student. The phenotypic ratio, and the ratio is not discussed in your textbook, but at the same time, polygenic inheritance is very important. For example, capital A, capital A, capital B, capital B, capital C, capital C. Is it one gene? No. Is it two gene? No. And it is three gene, many gene, one character. Opposite of pleiotropy, it is called polygenic inheritance. And here it is crossing with the small a, small b, small a, small a, small b, small b, small c, small c. We are getting a gamete called capital A, small a, capital B, small b, capital C, small c. And how many gametes will be there, my dear students? 8 gametes. How I am getting the number 8, my dear students? How can you find out 8 gametes? There is an equation is there, and that the equation is 2 raised to n. What is 2 raised to n, my dear student? What is n actually? N is the total number of heterozygous condition. What is heterozygous condition? What is heterozygous condition? I will give. So here this is a heterozygous condition. This is a heterozygous condition. This is a heterozygous condition. Before that, and is there any heterozygous condition here? No. So that's why 2 raised to 0. What is 2 raised to 0? 2 raised to 0 is 1. So that's why, and here capital A, capital A, capital B, capital B, capital C, 2 raised to 0, that's why 1. That's why there is only one gamete. But here you can see it is 2 raised to, 2 raised to 1 heterozygous condition, second heterozygous condition, third heterozygous condition. So how many heterozygous condition? 3. So that's why 2 raised to 3, 2 raised to 3, 2 into 2 into 2, 2 into 2, 4, 4 into 2, 8. That's why the reason 8 gametes. So 8 gametes are produced, my dear student, and under this particular category. The question is not the gametes. If one parent can produce 8 gametes, the other parent also can produce 8 gametes. So 8 into 8. 8 into 8, my dear students. So that's why the total number, there are 64 young ones are produced, my dear students. So far we studied monohybrid and dihybrid. First we studied 4. Next one we studied 16. No, in a polygenic inheritance, total 64. And what is the ratio, my dear student? The ratio is not given in the textbook, my dear students. So please, you just note it down. The skin color in human being. Skin color in human being is the best example for polygenic inheritance. There are 64 boxes are there in the F2 generation, and the ratio is 1:1 to 16 is to 15 is to 20 is to 15 is to 6 is to 1. I will repeat one more time. 1:6 is to 15 is to 20 is to 15 is to 6 is to 1. So you know that 20, 15 plus 15, 30. So 20 plus 30, 50, 56, 62, 63, 64. The total sum must be 64. It is 64 itself. This is the ratio, my dear students. So 1:6 is to 15 is to 20 is to 15 is to 6 is to 1 is the ratio. And that is the answer. What is the answer? The phenotypic ratio. And the phenotypic ratio is 1:6 is to 15 is to 20 is to 15 is to 6 is to 1. That's about the phenotypic ratio. Okay. Moving into the next question. Here you can see, my dear students, in a honeybee colony. Oh, it's an important question, my dear student. Sex determination. The next topic is sex determination. In sex determination, drones do not have father. Oh, that is a important things, right? Drones are, what are drones actually? In a honeybee colony, my dear student, drones are the males. Males are called the male, honeybees are called the drones, you can see. So the drones are not having father, as well as drones are not having son. Drones are having mother, drones are having daughters. I will repeat, drones are having mother, drones are having daughters, but drones are not having father and drones are not having son. But they are having grandfather and they are having grandson. Oh, that's a unique feature, right? That is a sex determination. And you know, that is one of the important feature, my dear student, in sex determination in honeybee. Sex determination honeybee. You can see a picture, my dear students. Okay, and here you can see this is the queen, here you can see the female, and here you can see the drone, you can see. Here how many chromosomes are there, and here you can see 32 chromosomes are there in females, but only 16 chromosomes are there in male. Generally, the sex determination, you study different other type of sex determination. XXXO sex determination, XXXY sex determination, ZZZW sex determination, ZZZO sex determination. These are the 4 sex determination you studied. In that you can see it is not haploid diploid condition, and the number of sex chromosome in XXXY type, and you know, coming under human being and Drosophila, and that method is human males are determining the sex of the child, right? In humans, you can see the father is determining the sex of the child. Mother has no role in determining the sex of the child. Okay, and here you can see here 32, 16, here 32, here you can see gamete 16, 16 and 16, 16. Here also 16, 16, and it is mitosis. This is meiosis. Meiosis means reductional. Mitosis means equational. And here you can see what will happen. And you know, the drones, right? This is the gamete. So when both combine, look at the board. Here you can see this 16 and this 16 combined. You can see a female is produced. Female means what? The daughter, right? But here you can see, what about, you know, the drone cannot make his son, because, you know, already 16 chromosomes are there, and parthenogenesis is not reported. Parthenogenesis is not reported in haploid sperm. Parthenogenesis is there in the female gamete. Here they undergo a process called parthenogenesis, and they are undergoing a process called parthenogenesis, and directly from the female gamete 16, a young one is produced. This is the male, my dear student. So the male is having only the mother. The male is having only the mother. The male is having only the mother. The male is having only the daughters, but there is no father and there is no son. But you know, grandson is there. No, if this is the mother, she can produce young one, right? So that's why, and son will be produced. So this is the father. Father means, so this is the grandfather of that child. So that's why mother's father. That's why grandfather is there. I like that grandson is also there. And this is the son means what? The son is fertilizing with the female gamete to make a female. So that's why you can see, as well as son is also there in parthenogenesis. So altogether we can say that grandson is there, grandfather is there. son is not there, father is not there. Okay, so that is the question here. You can see justify the statement using a suitable diagram. The diagram is there in the textbook, but the textbook diagram is inadequate, my dear students, because just giving an idea how many chromosomes are there in male, how many chromosomes are there in female. Okay, and for your board examination, that is enough. Okay, the next one. Actually, there is something related with the pedigree analysis, my dear student. And here you can see the following pedigree shows a particular trait. Here you can see, that all the symbols you are very familiar with, male, female, and the parents are not having any genetic disorder, but you can see they are having 1, 2, 3, 4, 5. 5 children are there, and you can see the second child is infected, and the fourth child is also infected. And here you can see irrespective of the sex. So that's why it is not a sex-linked disease, it is an autosomal. So first of all, we can identify it is autosomal, my dear students. Autosomal. So here the question is, the following pedigree shows a particular trait which is absent in the parents. Actually, that trait is absent in the parents. Absent in the parents means what? They are, if it is a dominant trait, you can see, and that character is there in parents, but it is a recessive one. So recessive traits are not there in the parents, my dear students. So that's why, and they are heterozygous, and they are not having that trait. So that's why they are autosomal as well as, and they are recessive, my dear students. And here, but found in the subsequent generation, irrespective of the sex, so that's why it is not sex linked. Okay, and here analyze the pedigree and draw a conclusion. This is the conclusion, my dear students. What is that? And the trait which is depicted on this question is autosomal as well as recessive. And you can, along with that, you can write some example of autosomal recessive disorders, my dear student. And you give me some autosomal recessive disorders, and you already studied PKU, the phenylketonuria, the sickle cell anemia, the thalassemia are the 3 autosomal recessive disorders you studied. Okay, there are 2 sex-linked recessive disorders also you studied. What are the 2 sex-linked disorders you studied? One is called hemophilia, other one is called color blindness. And there is one autosomal dominant disorders also you studied, my dear student. Here the young one is having the disease, and one of the parent is also having the disease. That is called autosomal dominant, not recessive. In autosomal dominant case, myotonic dystrophy, muscular dystrophy, or myotonic dystrophy is the best example for autosomal dominant case. Okay, and the next question is related with the color blindness. And there are different types of color blindness, that. And when we are discussing the color blindness case, my dear students, that how can we, so we can see all the colors, right? And there are 3 different types of colors detecting cells are there in our retina, my dear students. Okay, and erythrolab, chlorolab, cyanolab. Erythrolab, chlorolab, cyanolab. Erythrolab can detect red color, chlorolab can detect green color, cyanolab can detect blue color, and using all other colors we are seeing today, are the combination of these 3 colors, my dear student. Your eyes can detect only 3 colors. Okay, and the remaining all are the combination of these 3 colors, my dear student. And here you can see a colorblind person, and colorblind person, you can see for driving test or something, colorblind person should be given a special type of chart, my dear student, and the chart is called, that word is also not there in the textbook. There may be chance for asking a question, Ishihara chart. What is Ishihara chart? And that the doctor is showing an Ishihara chart, and you are asked to say the number lies in that Ishihara chart. And if you are not a colorblind person, you are giving the right or the correct number. Otherwise, you are saying in another number, because if you are colorblind, you cannot get that particular number in the proper manner. The chart is called Ishihara chart. Whenever there is a question related with the color blindness in board examination, you can write the word Ishihara chart for detecting the color blindness, my dear student. Ishihara chart. And here you can see a female. Here the question. A female whose father is colorblind. A female, listen, female means what? What is that? X. And her father is colorblind. Colorblind means what? XCY. So this is, what is the question? A female whose father is colorblind. So that's why if her father is colorblind, you know that the Y is not given to her daughter, right? So that's why only X is given, means what? One X is XC. So a girl, he is having a colorblind father, means what? Colorblind father is having, come on, colorblind father means what? XCY, right? Hey, XC, C stands for color blindness. If it is hemophiliac, it is called XHY. Hemophiliac, it is XCY means what? Colorblind father. So a colorblind father can produce a daughter means what? A daughter is always having that XC gene. Hey, because you know the girl, you know the what is other X? Other X is from the mother, right? So a daughter is getting 2x, 1x from the father, 1x from a mother. So 1x from father means what? Father is already colorblind. So that's why you know XC. Okay, that is the conclusion you are getting from that. A female whose father is colorblind marries a male. Male means what? What you have to write? XY. Okay, a male whose father is also colorblind. So colorblind means what? XCY. But you know there is no problem for the husband. You can see the reason here. The father is XCY means what? X is not coming. X is coming only to the daughters. And for the son, only Y is coming. Is there any issue for the Y? There is no issue for the Y. And the problem is there only for the X. So that's why, and you know, here the normal male and here the carrier female. And what is the question if father is also colorblind? There will be a chance for such a question, my dear students. So you have to assume if father is colorblind, only X is given to the daughter. And if father is colorblind, only X is given means what? There is no problem for the son. What is the question? And you know what will be the possibility of their children to be colorblind? So that's why you have to do the cross, my dear student. How can you do the cross? Here you can see XX and a normal girl, XY a normal boy, and here you can see XCX a carrier daughter. And what is the last one, my dear student? XCY. So what is the question? What will be the possibility of their children to be colorblind? What is the possibility? Here the first one is not colorblind, second one is not colorblind, third one is not colorblind, the fourth one is colorblind means what is the possibility? 25% of their children, 1 by 4. 25% of their children are having the possibility to become colorblind, my dear student. That's a question. You can see the explain using a cross. This is a cross. Okay, and you know moving into the next one. Reasoning type question. It's a very easy question, my dear student. Reasoning type question. But I think one word I have to give one extra point. So that's why please listen to the board, my dear student. Hemophilia B is also called Christmas disease. So whenever you hear the word Christmas, December 25th, no, my dear students, you know, it is not that Christmas. Actually, you can see, and there are two different types of hemophilia. You are very familiar with the hemophilia. Hemophilia is a sex-linked, a X-linked, recessive disorder. Hemophilia is generally affected in boys only, males only. Because in female, so you know, male is having only one X chromosome. If that X chromosome is affected and you know the pair is Y chromosome cannot dominate over the X, right? So that's why you can see the XHY means the male is affected. But the female, you can see if one X chromosome is affected, XH only, but the other X chromosome is normal means what? XHX means what? She is not hemophiliac. The royal Queen family of what we can say the Queen Victoria family of UK in England. She is also a carrier. So that's why the reason this hemophilia disease is also called a royal disease. But the question is not royal disease. Why it's known as Christmas disease? Hemophilia is of two types, my dear students. Look at the board. It is not there in the textbook. That's why. Hemophilia, the first one is called hemophilia A and other one is called what? Hemophilia B. There are two different types of hemophilia. One is called hemophilia A, other one is called hemophilia B. Most of the cases we can see hemophilia A or hemophilia A is mostly occurring one. It is due to the deficiency of a factor named 8, my dear students, and you know 13 factors are there for clotting. You know that whenever you are having a bleeding, whenever a blood is moving out, automatically you can see the platelets are coming into that area and you know stops the bleeding, and that is what we call what the blood coagulation or blood clotting. There are 13 factors are there. Out of the 13 factors, you can see the 8th factor, if factor 8 is having a problem and that is leading into a clotting disorder, that is what we call what? Hemophilia A. And most of the hemophilia cases are hemophilia A. What is hemophilia B, my dear student? It is what we call what? Factor 9. Look at the board, my dear student. And if a person is having a problem with the factor 9, If a person is having a problem with the factor 9, and that is leading into a condition called what? Hemophilia B. So what are the two hemophilia? Hemophilia A, hemophilia B. Hemophilia A is due to the a disorder in factor 8, and hemophilia B is due to a disorder in factor 9. But still we are not reaching the answer. And why hemophilia B is called Christmas disease? As you all know that, my dear students, here you can see the factor 8. The factor 8, look at the board please. The factor 8 was first reported from a patient named factor 9 related hemophilia B. Factor 9 related hemophilia B was first identified in a person named Stephen Christmas. So Stephen Christmas is the name of the patient. So the first a patient name, that is Stephen Christmas. So that's why the reason factor 9 is called Christmas factor. And that's why the reason the hemophilia B is also called a Christmas disease. Okay, so that's why the reason the Christmas disease, my dear student. Okay, the next one, GLU is replaced by VAL in SCD. First of all, you should know that what are the full form of that particular terms, my dear student. What is GLU? As you all know that it is a type of amino acid. What is amino acid actually? It is what we call glutamic acid. And there is an amino acid called glutamic acid, my dear students. And glutamic acid, as you all know that, we studied, what is the disease actually? It is called, what is SCD? It is sickle cell disease, sickle cell anemia. And sickle cell anemia, we are not using the term SCA, we are using SCD, sickle cell diseases. And the sickle cell anemia is due to, you study, there is one of the important mutation. You study, what is that mutation called? What? The mutation is called point mutation. What is point mutation, my dear student? What is point mutation? What is that? And adenine and thymine is having an inversion. You can say point mutation. Due to that point mutation, in the 6th position of the beta globin chain, the normal one, glutamic acid, is replaced by another one called what? Valine. Instead of GAG, it is GUG. If it is GAG, it will be glutamic acid. Instead of GAG, the mRNA is having a triplet code, a genetic code called GUG. If it is GAG, it can make the glutamic acid. If it is GUG, it can make valine, and that is leading into a condition called sickle cell disease, and it is an example for point mutation. Mention any two difference between the two types of thalassemia. And you know that, there are two different types of thalassemia. Look at the board, my dear student, the thalassemia. So, we are covering almost all the portions. So that is why the reason each and every topic is included. Please listen, please. And here you can see, this is what we call what? Thalassemia, my dear student. Thalassemia. Thalassemia is also an autosomal recessive disorder, which is a Mendelian disorder. And the thalassemia, there are two different types of thalassemia. You're very familiar with. One is called alpha thalassemia. One is called alpha thalassemia, and other one is called what? Beta thalassemia. So chance were asking a question, my dear student, what is the difference between sickle cell anemia and thalassemia? Sickle cell anemia and thalassemia, what is the similarity? Both are autosomal, recessive disorder. At the same time, that what is that? These two, one is the sickle cell anemia is a type of qualitative disorder, and you can see this is a quantitative disorder. Because what is the difference? Look at the board, please. What is the difference here? Here you can see alpha thalassemia. The problem lies in chromosome number 16. The thalassemia, the issue, the alpha thalassemia, the issue is there in chromosome number 16, and beta thalassemia, it is chromosome number 11. Whenever there is any question, difference between, whenever there is a question, differentiate between, if there is any such a question, it is your responsibility to draw a table with two columns. In board examination, presenting the paper in your best method, that also matters, my dear student. Because the last 1 year you studied a lot of different things in bio, but you can see you are getting only 3 hours. You are getting only 3 hours to showcase your brilliance. You are getting only 3 hours. The whole 1 year, 365 days you studied, but you are getting only 3 hours. Only 3 hours to show your brilliance means what? The presentation matters. So you should make a table with the 2 columns, and the first column you can write alpha thalassemia, and the second column you can write beta thalassemia. And the first point you can write, it is called what? It's a chromosome number 16, and here you can see chromosome number 11. And you know how many genes? One more difference, because they asked 2 differences. So here you can write what? Here you can write, look at the board, my dear student. Here you can write, there is a gene called HBA. HBA gene, and here also HBA2. There are 2 genes. HB stands for hemoglobin, A stands for alpha, 1 stands for the first gene. HB hemoglobin, A stands for alpha, 2 means the second gene. So HBA1, HBA2. And how many genes? There are 4 genes. Why 4 genes? I wrote only 2 right here. Hey, I wrote only 2 here, HBA1, HBA2. And then why I use the word 4? Because one is from paternal, other one from maternal. Each one pairs are the right. And you are getting HBA1 from father, HBA1 from mother, HBA2 from father, HBA2 from mother. So total 4. And here you can see, here there is only 1 gene is there. That's why there is no number. HB stands for hemoglobin, B stands for beta, HBB in chromosome number 11. The same chromosome having the problem sickle cell anemia too. The reason, beta globin, this is alpha globin. A hemoglobin consists of 4 chains, 2 alpha chains and 2 beta chains. So these are the 2 major differences between alpha thalassemia and beta thalassemia. It's a quantitative disorder, and the quantity of hemoglobin matters more less the quantity, and more the issues. Okay, that's about the question. Okay, moving into the next one. Name any 2 types of aneuploidy. Name any 2 types of aneuploidy. And you already studied, after the mental health disorders, in the last topic of the chapter you studied. What is that? There are 2 different types of disorders. One is called what? Aneuploidy. Important topic. You can expect a question from this topic, aneuploidy. And other one is called what? Polyploidy, my dear students. One is called aneuploidy, another one is called polyploidy. What is aneuploidy? The definition is very important. What is aneuploidy? The segregation. Listen, but generally segregation is taking place. And what is aneuploidy? Failure of segregation. So failure of segregation of chromatids. Hey, and that sentence is very important. Failure of segregation of chromatids during the meiotic division. Failure of segregation of chromatids. What is a polyploidy? And it is a failure of cytokinesis. It is a failure of cytokinesis. Both are totally different. Here, polyploidy, failure of cytokinesis means what? It is making either 3N or 4N, either triploid or tetraploid. Triploid or tetraploid. Triploid condition and tetraploid condition is not there in human. That is only in plants. That's why polyploidy is reported only in plants. But what about here, my dear students? Aneuploidy is reported in humans, right? And aneuploidy is a failure of segregation means what? Here, aneuploidy, there are 2 different types of aneuploidy. There are 2 different types of aneuploidy. What are they? Here the same question. Name any 2 types of aneuploidy with a suitable example. Here you can see one is called monosomy and other one is called what? Trisomy. And there are 2 disorders, my dear student. One is called monosomy and another one is called trisomy. What is monosomy, my dear student? At 2N minus 1. What is a 2N? 2N means what? 46 chromosomes. 2N means diploid 46 chromosome, right? So that's why monosomy is producing the chromosome number 45. And here you can see trisomy means what? 2N plus 1. So that's why the chromosome number 46 plus 1, that is 47. If your chromosome number is 46, there is no chromosomal disorders. Chromosomal disorders either due to the deletion of 1 chromosome or due to the addition of 1 chromosome. Deletion is called monosomy, addition is called trisomy. And here you can see, and that is what we call monosomy and trisomy. And what are the examples, my dear student? What are the examples you studied, please? What are the examples you studied? Here, monosomy, you studied one example called a Turner syndrome. Turner syndrome, you studied. What is Turner syndrome? A female is produced without the feminine characters. A female is produced without the feminine characters means what? A female is having rudimentary ovary, breast development is not there, and the development of ovum is not there. So altogether you can see looking like a female but without the feminine female characters. That's called a Turner syndrome, XO condition. You studied, no, XO. It is not O actually, it is 0, and it is not XX. Normal girls are having XX, right? Here X and a gap. That gap is represented by O or 0, XO type. Trisomy. There are two examples, my dear student. One is called a Down syndrome. One is called a Down syndrome, and other one is called a Klinefelter syndrome. And you know, a Down syndrome is an autosomal chromosomal disorder. So that's why it is also called a 21 trisomy. Why it is called a 21 trisomy? In the 21st chromosome, instead of 2, there are 3 chromosomes are there, and that is what we call what a autosomal issue. That's why both males and females are affected, my dear student. And what is the last one? Klinefelter syndrome. What do you mean Klinefelters? It is called XXY. Generally, a boy is having XY, a girl is having XX, but here you can see one chromosome is additional, sex chromosome is additional, XXY, that is sex chromosomal. And here you can see that sex chromosomal XXY, and it's a boy or a girl. It is a boy at the same time with the female characters. A boy with the female character, and the boy is developing the breast, you know that. A boy with the breast, it is called a gynecomastia. Gynecomastia. What is gynecomastia? Textbook word, NCRT textbook word itself. Breast development in boys. Breast development in girl is not a disorder, but breast development in boy is a disorder named gynecomastia, reported in Klinefelter syndrome. The Klinefelters and Down syndrome together called the trisomy case. So this is the question. Name 2 types of aneuploidy, monosomy, trisomy. One example each. Here we wrote 1 example. Here we wrote 2 example. That's about the chapter principles of inheritance and variation. Okay, so the first chapter we completed, my dear students, and within 5 minutes, within a very short break, we can move to the next chapter, molecular basis of inheritance. Please be there, please. Hi, welcome back, my dear students. Actually, the last session we were discussing about the chapter Principles of Inheritance Variation. The first chapter in genetics, almost 8 or 9 marks we are expecting for the coming examination. And you know the next chapter, molecular base of inheritance, that is equally weighted chapter, that also we are expecting the same 8 or 9 marks. And you know that the unit 2, the second unit, genetics and evolution, a total of 20 marks. Out of the 20 marks, you can see both principles and molecular bases both are having nearly 15 or 16 marks, my dear student, and the remaining 4 marks from the topic evolution. So here you can see we are moving into the next chapter, molecular base of inheritance, some important concepts. So all of you must give proper response, my dear students, because these 2 chapters are very important for your board examination. Okay, on March 19th, Tuesday. Listen please, the first question. And you know in the chapter you can see what is the molecular basis of inheritance actually. The chapter name is the molecular base of inheritance, and what is known as the molecular base of inheritance? It is nothing but DNA. And here there is a question, you know, it is related with the length of DNA. So, you know, there are different lengths are given here. You can see bases are there as well as base pairs are there. Bases means single-stranded DNA and base pairs means double-stranded DNA. So here you can see that, and what are the topics given, what are the concepts given in your textbook. You can see the length of DNA, the length of the molecular base of inheritance, what is, and here you can see there is a special type of bacteriophage, a virus 5x174. And you can see the 5x174 is a type of bacteriophage with you can see 5,386 bases are there. So that's why they are having single-stranded DNA. So it is having a single-stranded DNA, so that's why it is not base pair, it is bases. 5,386 bases are there, my dear students. And what is the next one? The next one is called bacteriophage lambda, and the bacteriophage lambda is having 48,502 base pairs are there. That's why it is double-stranded DNA. So always you keep your mind, the number is very important. At the same time, bases or base pair, that is also matters. Okay, so altogether you can see the 5x174, a bacteriophage with 5,386 bases, only single-stranded DNA, and bacteriophage lambda with the 48,502 base pairs, that is a double-stranded DNA. You can see the E. coli, the most common term in NCERT textbook, the bacteria E. coli, that is almost 4.6 into 10 raised to 6 base pairs are there in E. coli. While you can see in humans, you can see in a haploid content, that is 3.3 into 10 raised to 9 base pairs, and this is in haploid condition. If it is in diploid condition, that is 6.6 into 10 raised to 9 base pairs. And these are the 4 data given in the textbook, my dear students, and 2 bacteriophage and 1 prokaryote, other one is a eukaryote human beings. Okay, so using this data, you can easily answer this question. The length of DNA in a bacteriophage lambda. In bacteriophage lambda, it is 48,502. But there are 2 answers, 48,502, 48,502. But you know the bacteriophage lambda is having double-stranded DNA, so that's why it is base pair. That's why D is the right answer. Moving into the next question. In a double-stranded DNA, so you know the DNA related some questions. What are the questions you have to study under DNA, my dear students? And you know DNA as an acidic substance. The first, that concept was developed by Frederick Meischer, my dear student. Frederick Meischer discovered an acidic substance in nucleus, my dear student, and he named it as nuclein. And you know, he had no idea about the DNA, so that's why he put a name, nuclein. And later you can see the X-ray diffraction model given by Wilkins and Franklin to explain the structure of a double helical model. Unfortunately, that also was not a successful move. But based on the X-ray diffraction model by Franklin and Wilkins, you can see in 1953, the James Watson and Francis Crick together proposed the double helical model of DNA. The double helical model of DNA, there are some salient features are there. Along with the salient features, you have to study the base parity rule given by Erwin Chargaff, the Chargaff rule. And in a double-stranded DNA, look at the board, in a double-stranded DNA, and what is the Chargaff rule actually? What is Chargaff rule? And the number of adenine plus guanine is always equal to the number of thymine and cytosine. What is adenine? Adenine is a purine, my dear student. What is guanine? Guanine is also a purine, my dear students. And thymine is a pyrimidine, and cytosine is also a pyrimidine. How can you know that it's a purine or pyrimidine? And purines are always, look at the board, my dear student. Purines are always, you can see, double ringed structures. And purines are double ringed structure, while pyrimidines are single ringed structures. Okay, and this is the difference between purines and pyrimidines. And what is A plus G? So why A plus G is equal to T plus C, my dear student? Simply we can say that A is always combining with the thymine, and G is always combining with the cytosine. Adenine combines with the thymine using double hydrogen bond, and guanine combines with the cytosine with the triple hydrogen bond. So that is why the reason, if look at the board please, if the number of adenine is 120, as you all know that, what will be the number of thymine? The number of thymine is also 120, because adenine can pair with only with the thymine. If the number of G is 80, my dear student, the number of cytosine is also 80, my dear students. And why? Because guanine can combine only with the cytosine, unless if it is a double stranded condition. In single stranded, this rule, the Chargaff rule, the base parity rule, is not applicable in single stranded DNA. It is applicable only in double stranded DNA, my dear students. And here you can see, so that's why easily you can say 120 plus 80, 200, 120 plus 80, 200. So that's why A plus G is equal to T plus C is the first rule given by Erwin Chargaff, textbook content. And at the same time, you can see there is one more rule is there, my dear student. It is A plus G by T plus C is always equal to 1. This is also given in your textbook. What is A plus G? The total number of purines. And what is T plus C? The total number of pyrimidines. The total number of purines by total number of pyrimidines always is equal to 1. Here 120 over 80, 120 plus 20, 80, 200, 200 by 200, it is equal to 1. And this rule is applicable only in double-stranded DNA, given by Erwin Chargaff. Okay, so that's why the reason, what is the answer for this question? In a double-stranded DNA, adenine, the number of adenine, not the number is given, percentage is given, and the number of adenine is 35%, my dear student. And here, what will be the percentage of the other purine? There is one more purine, no. What is other purine? The other purine is guanine. You can see adenine and guanine are purines. What is the percentage of the other purine? So, how can you find out the answer? If adenine is 35, what will be the thymine, my dear student? Thymine also will be 35%. Why? Why 35%? Because adenine 35 means, thymine is also 35, same number, right? So that's why, 35 plus 35, 70%. Out of the total 100%, 70% is occupied by adenine and thymine. And what is remaining, my dear student? The remaining only 30%. And but that 30% is equally divided into guanine as well as cytosine, my dear students. Okay, so equally dividing into guanine and cytosine means what? Guanine 30 divided by 2. So here it will be 15%, and here also it will be 15%. That's why 35 plus 35 plus 15 plus 15, 35 plus 35, 70%, 15 plus 15, 30%, 70 plus 30, 100%. So that's why the guanine is 15%. Answer, the first one is wrong, second one is wrong, third one is wrong, because 15% is not given. So that's why the answer is none of this. Moving into the next question, my dear students, moving into the next question. What do you mean by feminism? Actually, in your textbook, you can see in italics that a question is asked, you can see. So all of you look at the board, I will explain you the concept of feminism. Before discussing feminism, you must have an idea about what is the central dogma in molecular biology. What is the central dogma in molecular biology, my dear student? The central dogma in molecular biology is nothing but the protein synthesis. And you're very familiar with the process. All of you look at the board. What you can see, there is a double-stranded DNA. So I am not discussing terminism now. I'm not discussing terminism now. I'm discussing the central dogma, because central dogma reverse is called terminism. So that's why, what is central dogma? So for the time being, we can discuss the first one, that is central dogma. And my dear students, central dogma, double-stranded DNA is changing into double-stranded DNA. That is called DNA replication, the first one. And the double-stranded DNA is changing into single-stranded mRNA, and that process is called transcription. And after that, you can see the single-stranded mRNA undergoes translation to make a polypeptide, and that is what we called what protein. You can see this process take place in nucleus. This process also take place in nucleus, while the translation process take place in cytoplasm. And the diagram what I represented on the board, and this is what we called what the central dogma in molecular biology. The concept was given by Francis Crick. So Francis, the same, James Watson and Francis Crick, right? The same Francis Crick developed the concept central dogma in molecular biology. This is applicable in a case by which the genetic material should be DNA. But you studied, no, my dear student, there are so many viruses are there, especially the retroviruses. You can see, you already studied some retroviruses or viruses with RNA as the genetic material. Tobacco mosaic virus, a QB beta virus. In your textbook, it is given QB means what? QB. There is a topic, is there? No, in molecular biology. What is the DNA versus RNA? It is given a special type of bacteriophage called QB bacteriophage. What is the full form of QB bacteriophage? Q stands for QB and B stands for beta. So QB bacteriophage means what? QB beta bacteriophage, and that is having RNA as the genetic material. And here you can see the retrovirus, RNA is the genetic material means what? If RNA is the genetic material, the central dogma is not in a progressive manner, it is in a reverse direction. So that's why RNA to DNA. RNA to DNA. That concept is called what? Reverse transcription, my dear student. And reverse transcription means what? This is central dogma, and this is what we call a central dogma reverse. And central dogma reverse means what? It is not the concept developed by Francis Crick. And that central dogma reverse, that concept is developed by two scientists. One is called Baltimore, and other one is called Temin. And you know, there are Temin and Baltimore, or are the two scientists, Temin and Baltimore. So in memory of that Temin, you can see we are using the term terminism. So in your textbook, it is given the central dogma reverse in RNA viruses, COVID-19 viruses, the human immunodeficiency virus, HIV that causes AIDS. Altogether, we can say that it is terminism, central dogma reverse. And if there is any question related with the central dogma reverse, my dear student, you can add this additional point, terminism, the concept developed by Temin. At least you have to study Temin, no need to study Baltimore. And the central dogma reverse, RNA to DNA, that is reverse transcription, terminism. Important point. Next one. The total length of DNA in E. coli is, oh, it's an important question. Listen please, my dear students. Here you can see the total length of DNA in E. coli. The total length of DNA in E. coli is 1.36 millimeter. Okay, so 1.36 millimeter. 1.36 millimeter. It is what is that? 1.36 millimeter, and that is 1.36 into 10 raised to minus 3 meter. And here you can see, find out the total number of base pairs in the DNA. So that's why the total length is always equal to the total number of base pairs into, listen, the total length of DNA is equal to total number of base pairs into the distance between the base pairs. This is the equation, my dear student. The total length of DNA is equal to base pairs, the total number of base pairs into distance between the base pair. You know that we can take humans into account first. First of all, we can study humans into account. And the humans, how can we find out the total length, my dear student? What is the total number of base pair? 6.6 into 10 raised to 9 base pairs. And what is the distance between the base pair? That also you studied, 0.34. And that is in nanometer. How can we convert nanometer into meter? 0.34 into 10 raised to minus 9 meter. Okay, I will repeat. The total number of base pairs 6.6 into 10 raised to 9 base pair, and the distance between the base pair, that is 0.34 nanometer, 0.34 nanometer, 0.34 into 10 raised to minus 9. And you know, when you are adding this one, what is that? A raised to m into a raised to n. You studied a raised to m plus n. So 10 raised to 9 minus 9, that is gone. So that's why 6.6 into 0.34, that is 2.244 meter is the total length of DNA in human. In your textbook only the answer is given. This is the method, my dear student. The total number 6.6 into 10 raised to 9 base pair. The distance between each base pair is 0.34 into 10 raised to minus 9. When we are multiplying both, you will be getting 2.244 meter. In your textbook it is given 2.2 meter, and the real answer is 2.244 meter. It is the total length of DNA in humans. And here the question is not human, but you know, as a part of your revision, I have to explain about the other topics too. And here you can see the total length of DNA. Look at the board, please. Here you can see it is 1.36 millimeter is equal to 1.36 into 10 raised to minus 3. So here the length is given, my dear student. You have to find out the base pairs. So base pair into a distance, my dear students. How can we find out the base pair, my dear students? What is the length actually? 1.36 into 10 raised to minus 3 divided by. What is the distance actually? It's multiplied by, that is for the other one. Here you can see 0.34 into 10 raised to minus 9 meter. Okay, so that's why minus 3 minus, so a raised to m by a raised to n. So a raised to m minus, so a raised to m minus n. So that's why 10 raised to minus 3 minus 9. So that's why it is 10 raised to 6. So here you can see the answer will be 4 into 10 raised to 6 base pair. In the beginning we already studied 4.6 into 10 raised to 6. Both are correct. Okay, so 4 into 10 raised to 6 base pair, that is the answer. What is that? Total number of base pair, that is 4 into 10 raised to 6 base pair. That much number of base pairs are there in DNA. So that's about what we can say the question. So how can you find out the total length of DNA? That always you keep in your mind. Sometimes you can see there may be chance for asking an additional question other than human DNA and the E. coli DNA. And the answer is like this, my dear student. What is the total length of DNA? How can you find out? The total length of DNA is always equal to the total number of base pairs into the distance between the neighboring base pair. If it is given in millimeter, you have to convert it into meter. If it is given in nanometer, you have to convert it into meter. Then you have to do the procedure what we explained here. Okay, moving into the next question. Histone protein, as you all know that histone protein means what? You know the DNA, the length of DNA in human body, it is almost, you can see the 2.244 meter DNA. How can we afford this much lengthy DNA inside the very, very, very small nucleus, my dear student? So there should be a well-defined packaging of DNA is very essential in human body, or especially in eukaryotes. Prokaryotes not an issue because nucleus is not having a nuclear membrane, and it is the structure is called what? The nucleoid. The nucleus or the DNA is not covered by the naked DNA. The DNA is not covered by any nuclear membrane, my dear students. But in humans, you can see it is well covered by a nuclear membrane, means what? The DNA must be fully accommodated inside the nucleus itself. In that case, you know what we want to do? The histone proteins are the proteins, and histone protein is an histone octamer, you know that. And octamer means what? 8 units are there, my dear student. What are the 8 units? In your textbook you can see 8 molecules are there, 8 units are there together called histone octamer. But the number of, or what are the different 8 units are not given in the textbook. That also you should remember for the examination, because whenever there is any question related with the histone octamer, you can write this point, my dear student. And how many units? 8 units. What are they? One unit is there called H. H stands for histone H2A, one unit, and another one H2B, and another one H3, and the last one H4. I mentioned only 4, right? But you know all are in pair. H2A 1 pair, H2B 1 pair, H3 1 pair, my dear students, and the last one H4 1 pair. A total of 8 pairs. sorry, a total of 4 pairs, that is 8 in number, my dear student. So here you can see H2A, 2H2A, 2H2B, 2H3, 2H4, total 8 units, 8 molecules called what? Histone octamer. So here the question is, histone protein is made up of 8 molecules, and these are the 8 molecules you can see, and they are together termed as histone octamer. Octamer means what? 8 units. Octa means 8, mer means unit. What are the molecules? And here I already mentioned, these are the molecules. In your textbook you can see, if this is one histone and this is another histone, and how is the arrangement of DNA? The DNA is coming like this. Look at the board. The DNA is coming. DNA is negatively charged and histone is positively charged, and the histone is having two different types of amino acid. You can see lysine and arginine. It is important, the histone is having an amino acid called lysine, and other one is called arginine. Both are responsible for making the positive charge, because histone is highly positively charged molecule, my dear student. And here you can see the DNA is wrapping around the histone, and one more time it is wrapping around the histone, and after that it is moving out, and it is coming as, and it is moving into this side and wrapping around this one, and here wrapping around this one, and after that you can see it is moving like this. So this is what we call a nucleosome. This is called a nucleosome, and this is what we call a beads on a string like structure, and that is what we call a chromatin. And in some area chromatin is tightly packed to called euchromatin, which is light colored as well as transcriptionally active, and the other area of the nucleus by which the chromatin is densely packed dark in color, and that is transcriptionally inactive, my dear student. And here that is not the question, but that is also a topic of discussion, my dear students. Okay, moving into the next question. Who revealed the biochemical nature of transforming principle? You already studied the transforming principle, my dear student. What is transforming principle? And you can see, you studied the Frederick Griffith in 1928. In 1928, the Frederick Griffith introduced a particular concept called what? Transforming principle. He conducted experiments in mice, you can see, as well as he used two different strains of Streptococcus pneumonia bacteria that can cause pneumonia. R strain, the rough strain, S strain, the smooth strain, my dear student. And that two strain, which one is virulent, which one is non-virulent? As you all know that S strain is virulent because it can make a smooth mucus polysaccharide coat over that particular bacteria. So that is why the reason S strain is always virulent, my dear student. And in that biochemical characterization means what? Their last step is important, and here you can see the R strain is non-virulent, and another one heat killed S strain, and both are non-virulent, and if they injected into the mice, you can see, and this is R strain is non-virulent, and it's non-virulent means what? Not infectious, non-infectious. Okay, R strain is non-virulent. Heat killed S strain is also non-virulent, and both are injected into the mice means what? It is making, we are all expecting a result, mice live, but instead mice die, my dear students. And why mice die? Here you can see, and in the dead body you can see they obtained S strain, but S strain is not given, right? S strain is not given, right? S strain is not given by Frederick Griffith, right? Why? Because the R strain undergo transformation. R strain undergoes transformation to make a strain. So what is transformation? The genetic material inside the R strain transformed to form a strain, and that is responsible for making it virulent. So that is why the reason S strain is there. And this is the last step. This step you can see is continued by, the biochemical characterization. The biochemical characterization was given by who are the three scientists, my dear student? The name of the scientist is very important, my dear student, and called Avery. And there are three scientists together conducted the biochemical characterization of transformation principle developed by Frederick Griffith. The first person is called what? Avery. The other one is called what? MacLeod. And you can see the second scientist is called what? The MacLeod. And the third scientist is called what? McCarthy, my dear student. And these are the three scientists. Avery, MacLeod, and McCarthy were the three scientists who gave the experimental. So who revealed biochemical nature means what? There are three scientists, Avery, MacLeod, and McCarthy. So these are the three scientists who revealed the biochemical characterization. So you have to write what is that method actually? What is that method? They used one enzyme, RNase. When they use RNAs, all the RNA get completely digested, but transformation occurred. Transformation occurred means what? RNA is not the genetic material. They used another enzyme, protease, my dear student. They used a protease. All the proteins are digested inside the DNA, and all the proteins are digested. So transformation occurred means what? Proteins are not the genetic material. And finally, they add one enzyme, DNase, my dear student. What is DNase? Deoxyribonuclease. Deoxyribonuclease enzyme used means what? All the DNA get completely damaged, my student. All the DNA get completely damaged means what? There is no transformation. So no DNA, no transformation. No DNA, no transformation means what? DNA is the genetic material. That concept, the DNA is the genetic material, that concept was developed by Avery, MacLeod and McCarthy, and that is a biochemical characterization of transformation principle. But you can see the detailed concept was given by two scientists named Hershey and Chase. You can see the Hershey and Chase experiment you are very familiar with. In Hershey and Chase experiment, they proved that DNA is the genetic material and protein is not the genetic material using two radioactive isotope. One is called P32, other one is called S35. Why they selected P? Because P is present in DNA, absent in protein. Why they selected S35? They selected S because S is present in protein and S is absent in DNA. And that is Hershey and Chase experiment. That is also an important one. There is another question related with a replication, my dear student. There is chance for asking a 2 marks question. Draw a replication fork and label the parts. All of you look at the board. How can you draw a replication fork? Look at the board, please. A replication fork, how can you? So you have to start with, look at the board. Here you can see you are starting with a DNA strand. And here DNA strand, you can see their base pairs are there, my dear student. And the base pairs are there. And here one point is there, my dear student. The first point you have to label it as ORI. ORI, what is ORI? And in your textbook it is given in italics ORI, the full form of ORI. You can see, or not full form actually, ORI stands for, the ORI stands for origin of replication. It is the area by which the replication process begins, my dear student. So that's why the helicase enzyme binds in that area, and the remaining part of the DNA, here you can see one strand is there, here one strand is there, and this is what we call what? The 5' end, and this is called the 3' end. If it is 5' end, this is the 3' end. If it is a 3' end, this is the 5' end, my dear students. And here you can see this is the original replication by which you can see the DNA gets separated. And here you can see in this area, here you can see there are some unpaired bases are there: adenine, thymine, guanine, cytosine, thymine, adenine. You can write any bases here, and you have to write thymine. You are not supposed to write uracil because this is a DNA strand. And here in this strand you are not supposed to write another one. You have to write the complementary strand. If it is A, it will be T here. If it is A, if it is C, it is G, C, and here C, it will be G, my dear student, T, A, and if it is A, it will be T. And these are the two strands, my dear students. But this is the mechanism. But you can see the replication fork means what? Here you can see the enzyme is coming. Which enzyme? The DNA dependent DNA polymerase enzyme, which is working only in one direction, 5' to 3'. So that's why here when the enzyme is here, that is in 5' and it is moving into a 3' direction, my dear students. But that time they need some substrate, right? What is the substrate they need? Deoxyribonucleotides. And deoxyribonucleotides means what? And they are providing energy for DNA replication. That question came in one of the previous year question. And what are the dual functions of deoxyribonucleotides? What are the 4 different types of deoxyribonucleotide? Deoxyribonucleotide with adenine, deoxyribonucleotide with thymine, deoxyribonucleotide with guanine, deoxyribonucleotide with cytosine. So that's why first of all deoxyribonucleotide with thymine is coming and combining here. The first function, it acts as a substrate. At the same time, phosphate group is right. So that's why it can provide energy for the process replication. That are the 2 functions performed by deoxyribonucleotides. And here the next one is coming: adenine with double bond, guanine with triple bond, cytosine with triple bond, adenine with double bond, thymine with double bond. Here you can see this is a continuous strand, you can see. So this is what we call what? Continuous strand, my dear student. Continuous strand is also called one more name, and it is also called what? Leading strand. Continuous strand is also called what? Leading strand. But the other strand, you can see here also it is starting in 5 prime, but you can see when it moves in particular distance it is broken. So that's why 5 prime to 3 prime, here adenine double bond, here thymine double bond. Again 5 prime it is started and it ends in another area. So that's why here you can see it is guanine triple bond, cytosine triple bond. Again one more starting with the 5 prime and that ends in 3 prime, thymine adenine. Here you can see it is a continuous strand, right? Is it continuous or discontinuous strand? And this is what we called what? Discontinuous strand, my dear student. It is called a discontinuous strand, and there is a lagging because there are so many fragments are there, and these fragments are called what? Okazaki fragments, my dear student. It is called Okazaki fragments. The fragments produced in discontinuous strand, the fragments produced in lagging strand, which is called what? Okazaki fragment. And this is the diagram, my dear student. This is the diagram, the DNA, the replication fork. Label all the parts we labeled 5 prime to 3 prime, 3 prime to 5 prime, and the continuous or leading strand, discontinuous or lagging strand. These fragments are called Okazaki fragments, and this is the original replication. These are the parts you have to label in that particular diagram. That's about replication fork. Okay, and here the next question. Who proved DNA in chromosome is also responsible? And you know, in your textbook you can see there is one of the major experiment is given, right? What is that experiment? It is Michelson and Stoll experiment. Michelson and Stoll experiment, my dear student. Actually, what they tried to prove, my dear student, Michelson and Stoll. And Michelson and Stoll, as you all know that Michelson and Stoll, you know, they proved that DNA replication is semi-conservative. So, you know, they proved that DNA replication is semi-conservative. Actually, the concept DNA replication is semi-conservative, and that concept was developed by Francis and Crick. But you can see that concept was proved by Michelson and Stoll using N15, medium, and N14. And they are not the radioactive isotope, that is density gradient. N15 is a heavy isotope, and N14 is the light isotope. N15, the first one, and after 18 minutes or 20 minutes, you can see N15, N14, the hybrid is produced, and N14 is also produced. That experiment Michelson and Stoll, but that is not the question, because that is DNA replication only. Here the question is DNA in chromosome related. It is not Michelson and Stoll. It is not Watson and Crick, my dear student. And that answer to that question, it is textbook content itself, and that is Taylor and his colleagues. Taylor and his colleagues proved that DNA in chromosome is also replicated in a semi-conservative manner. And the other one, you know, the double helical DNA, right? The double helical DNA undergoes replication. That was proved by Michelson and Stoll using the major experiment, Michelson and Stoll experiment you studied. At the same time, at the last paragraph of Michelson and Stoll experiment, it is given that Taylor and his colleagues, you can see they proved that DNA in chromosome is also, important point to be noticed, DNA in chromosome is also replicated in a semi-conservative model, my dear student. And you know, name the experimental plant they are asking. And what is the, and you know, Taylor and colleague, the name of the scientist is not asked, but you have to study, because you know, for your board examination there may be chance for asking the Taylor and colleagues. And what is the name of the plant? The name of the plant is Vicia faber. And when you are writing scientific name, I already told, separate, underline, and it is commonly called what? Kidney beans or faba beans, my dear student. And it is what we call what? The Vicia faber, VICIA, Vicia faber, you can see. And you know what material they used? That is also very important. They used radioactive thymidine. So you know, in this area, my dear student, there are 3 points to be noticed. DNA replication in semi-conservative, that is Watson and Crick proposed, Michelson and Stoll proved. Here DNA in chromosome is also replicated in the same manner. That concept was proved by Taylor and his colleagues using radioactive thymidine in Vicia faber or faber beans. It is given in the textbook NCERT content. Question will be there from that topic. Okay, moving into the next one. Eukaryotic RNA polymerases. My dear students, you know the process transcription, you can see. The process transcription, you can see. There are 2 different types of transcription. One is called prokaryotic transcription, and other one is called what? Eukaryotic transcription. Prokaryotic transcription is comparatively very simple. What is that? And when DNA dependent RNA polymerase enters, there is a sigma factor for initiating the process, and there is a rho factor for terminating the process. And after the prokaryotic transcription, directly mRNA is produced. There is no intermediate structure developed. Directly the mRNA is produced. And what about here, my dear students? Here in eukaryotic, you can see instead of sigma and rho, there are so many factors are the transcription initiation factors as well as transcription termination factors. But at the same time, here you can see instead of mRNA, here in eukaryotes you studied hnRNA, heterogeneous nuclear. Heterogeneous nuclear. What do you mean by heterogeneous nuclear RNA? The DNA with the RNA with there are some areas are there called what? Introns. And there are some areas are there called introns, and other areas are called what? Exon. Exons are the expressing sequences, while you can see the introns are intron. Intervenous sequences are there. Intervenous sequence must be eliminated by a process called splicing, and after that 5-methyl guanosine is 5-methyl guanosine is added by the students. That is added in the 5-methyl guanosine is added in the 5' end. That is what we call what? Capping. And here a polyadenylate residues is added in the nearly 200 moieties are added. That is process is called what? Tailing. You can see. Then only we can call it as mRNA. hnRNA undergo processing or maturation to make mRNA. Okay, so I am not discussing that. Here the question is RNA polymerase. That is an important topic, my dear students. You listen. The RNA polymerase here, please. It is very important. RNA polymerases. In eukaryotes, my dear student, RNA polymerases are of 3 types. You can expect a question from that particular topic, my dear student. RNA polymerases are of 3 types, one by one. The first one, sure question. The first one, that is what we call what? RNA polymerase 1. The first one is RNA polymerase 1 in Roman letter. And RNA polymerase can make, all of you look at the board, my dear student, there are 3 different types of rRNA. What is that? The first one is called 28S rRNA. rRNA, what is that? Ribosomal RNA. The ribosomal RNA, there is a Swedberg unit, you can see sedimentation coefficient, you can see 28S rRNA. And the other one is called 18S rRNA. So 18S rRNA. And after that, the other one is called what? 5.8S rRNA. And these are the 3 types of rRNAs are produced. These are the 3 different types of rRNAs are produced from RNA polymerase 1. And after that, you can see the second one, you can see that is RNA polymerase 2. The second one is called what? RNA polymerase 2, which is very, very important, because RNA polymerase 2 can make hnRNA. You already studied hnRNA is the precursor of hnRNA is converting into mRNA, the messenger RNA, the most important one. But student, the mRNA is produced from hnRNA. hnRNA is produced from RNA polymerase 2, and hnRNA to mRNA splicing, capping, and tailing, my dear student, and that is RNA polymerase 2. And the last one, that is called what? RNA polymerase 3. And these 3 polymerases, most of the students are having a confusion over this category. That's why this is very, very important topic. RNA polymerase 1, there are 3. RNA polymerase 2, hnRNA to mRNA. And the last one, RNA polymerase 3, my dear student. RNA polymerase 3 means it can make, look at the board, RNA polymerase 3 can make an RNA called snRNA. What is snRNA? Small nuclear RNA. The small nuclear RNA. And other one is called what? tRNA, the transfer RNA. And one more, 5S rRNA. Don't confuse this one. This is 5S. This is 5.8S. This is 5.8S rRNA produced by RNA polymerase 1, but 5S rRNA produced by RNA polymerase 3. Okay, so this is what we call what the answer for that question. What do you mean by UTRs? The next question, what do you mean by UTRs? As you all know that in the topic translation you studied, what do you mean by UTR, my dear student? As you all know that when we are discussing the UTR, you must know the structure of mRNA. So, if this is the mRNA is a single-stranded structure, my dear student. You are not supposed to think it's a double-stranded one. It is a single-stranded mRNA, and the single-stranded mRNA, this is called 5' end, and this is called 3' end. All RNAs are only in one direction, that is 5' to 3'. And here you can see, my dear student, here you can see AUG. As you all know that AUG is the start codon, right? AUG is the start codon. At the same time, AUG can code for methionine. You know that. What is this one? UAA. You already studied, no? There are UAA, UGA, and UAG. UAA, UGA, and UAA, UG, and UAG are the 3 stop codons, and this is the stop codon, this is the start codon. In between, this is the area called the coding sequences. Between the start codon and stop codon, the area that is used for the coding sequences. Here you can see UUU. When AUG, that is leading into the formation of methionine. When UUU comes, that will be leading into the formation of phenylalanine. In your textbook, a checkerboard is there, no? When GAG comes, that is glutamic acid. When GAG comes, that is glutamic acid. When GUG comes, that is valine. When UAA comes, there is no amino acid stop codons. And this is what we call the coding sequence. But look at the board, my dear students. And what is this area? What is this area? Here you can see one area pointed out, and here also I am pointing out one area. These two areas are called what? UTRs, the untranslated regions. And that area is called untranslated region. To get the clear-cut idea, you can see the picture, my dear students. Here, in this picture, you can see different colors. You can see where is a 5' region. This is a 5' end of the mRNA that is shown in red color. And the 5' region, why it is given a cap, the word cap, because capping is taking place in that area. And what is the last one? Here you can see the 3' end. You can see the mRNA. Here it is given the poly A. What is that? A stands for polyadenylate. Polyadenylate residues are there in the 3' end, my dear student. The red color, the 5' end, the black color, the tail end you can see, and what is the green color region. And this is, here it is given the start codon, the AUG, and here the stop codon, UAA, UAG, and UGA are the stop codon, and the remaining area called what? Coding sequence. And what is this yellow color region? Important point, this topic, this diagram is not there, but you can see that explanation is given in your textbook, my dear student. And the yellow color region is called what? 5' UTR, 5' untranslated region. And here what is this color? And here you can see the 3' UTR. And what is that? And the 5' UTR is comparatively smaller than 3' UTR. This is the 3' UTR, my dear student. Whatever, there is no translation from 5' UTR. There is no translation from 3' UTR. Both the 5' UTR and 3' UTR together called untranslated region, UTR. That is a question asked. UTRs. Clear or not? What do you mean by UTR? Okay, so that is untranslated region. And the next question, draw a diagram to depict the lacoperon. That lacoperon, there are 2 diagrams are there in your textbook. One is in the presence of inducer, other one in the absence of inducer. Lacoperon, LAC means what? Lactose. Lactose is the inducer. Operon is the model. So lacoperon means what? Lactose operon, my dear students. Here you can see the question is by which inducer is present. Inducer is present means what? It is a system is switched on. So I'm going to draw the diagram. Look at the board. This is the lacoperon, and this is the lacoperon. Look at the board, my dear students. Here you can see, and it is polycistronic. It is lacoperon is there in E. coli. E. coli is coming under prokaryote. All prokaryotes are having polycistronic. What do you mean polycistronic? Is it one or more than one? More than one, right? 3 is right. So that's why this is the gene called Z. This is the gene called Y. This is the gene called A. These 3 together called what? This is 3 together called what? Structural gene, my dear student. Is it monocistronic or polycistronic? It is polycistronic. What about humans? What about human beings, my dear student? It is monocistronic. There is only one gene is there, monocistronic. Cistrone is having the capacity to make a protein, and here here polycistronic, many. So that's why here in just before this one, here you can see the promoter site is there, and in between promoter and the structural gene, there is a region, very important region. That is what we call what? Operator region. What is the name of that system? Lacoperon. Operon around the operator region. But at the same time, here you can see another promoter is there. Here you can see a gene is there, I gene, an inhibitor gene is there, and we are all having a thing in the lac operon inducer. I stands for inducer, no, I stands for inhibitor. It can make a repressor mRNA, a repressor mRNA can make a repressor protein. This is the repressor protein, but the question is inducer is present. If the inducer is absent, it is directly moving and blocking the operator region. If the system is switched off, the lac operon, the lactose is absent. But the question is, lactose is present. Inducer is present. That's why what will be the, and look at the board, my dear student, the inducer is coming. Look at the board here, you can see the inducer is coming, my dear student, and inducer is attaching with the lactose inducer. Only the lactose can be the inducer, and that lactose is attaching with the repressor, and the repressor is inactive, and that's why the repressor cannot move into that area. Automatically what will happen? These ZYA, the polycistronic gene, together make the mRNA, and from the Z, and there is chance for asking another question. From the Z, they are making an enzyme called beta galactosidase. Said gene can make beta galactosidase. MCQ type question. Beta galactosidase is produced by said gene or Y gene or A gene. The answer is said gene. But you can see the Y gene can make another one which is called permease, and permease, permeability related enzyme, permease you can see, and the A you can see, that is what we called trans-acetylase. And now the system is switch on. The system is switch on. Z is active, Y is active, A is active. Z can make beta galactosidase, Y can make permease, A can make trans-acetylase, and all these three enzymes together moving towards the lactose, and undergoing the digestion. Lactose is a disaccharide, right? It is digesting to form glucose and galactose. That is the diagram to depict lac operon system. Okay, I think it is clear, my dear student. Moving into the last question. What is the difference between southern blotting and northern blotting? So when we are reading that question, we are thinking southern and northern means what? Related with the north, south, west, east? No. It is not having any relationship with a south, north, nothing. And here you can see there is a scientist named E.M. Southern. A scientist named E.M. Southern developed a concept called what? Southern blotting technique, by which that is used for separating the DNA segments, my dear students. So DNA related separation. You studied one topic is there in the last page of your textbook of molecular base of inheritance, that is called DNA fingerprinting or DNA profiling with the VNTR variable number of tandem repeats, the repetitive DNA, the satellite DNA. In the topic, when we are discussing the steps of DNA fingerprinting, what is the isolation of genetic material, digestion by restriction endonuclease, separation by agarose gel electrophoresis, and after that we are moving into that the digested fragments are blotting. What is the method I use? Southern blotting, BLOTTING, blotting. Blotting means what? We are transferring it, my dear student. We are transferring the DNA segments into a nitrocellulose paper. That nitrocellulose membrane, that nitrocellulose paper, that method of transferring the DNA segments, that's why it is called what? Southern. Who developed that concept? E.M. Southern. That's why the mechanism southern blotting. After that, another group of scientists developed RNA segment separation. One scientist named E.M. Southern developed DNA segments, blotting of DNA segments called what? Southern blotting. Southern, the name of the scientist. And after some years, another group of scientists developed a method to transfer RNA segments. So that's why, what is the name given? If first one is Southern, automatically the next one is DNA and RNA, right? So that's why they put the name Northern. There is no scientist named Northern. The scientist is named Southern only. So Southern for, Southern for DNA and Southern for DNA segments, and Northern for you can see the RNA segments, my dear student. And that's about the last question, my dear students. Okay, so you know that here, when we are discussing the DNA fingerprinting, we discussed one mechanism, right? What is the mechanism we studied? The separated DNA fragments are transferred into agarose gel electrophoresis. What chapter it is? Is it the principles of inheritance and variation? No, my dear student, it is nothing but the biotechnology. And you know the Excel Education Center is coming with the chapters biotechnology 1 and 2 in day after tomorrow with the 2 chapters biotechnology principles and processes and biotechnology and its application and another YouTube live program. And thank you. Study well, my dear student. Best wishes. Thank you.